Good morning, biologists. Welcome to lecture. Today we're going to talk about the brain and the nervous system. I have a question for you. How much brain do you actually need to be able to function? And the answer is less than we expected. So Elise G, I'm showing you her MRI right here. She's missing her left temporal lobe. And the left temporal lobe, we've always thought was really important to language comprehension and reading. And so it was kind of amazing that she was missing this region of her brain because she scored at the top of every language assessment. She has this great quote, I could have probably taken over the world if I had my entire brain. And she didn't even know that she was missing part of her brain until she went in for an MRI for vision issues. So that's one example. Here's another example. In 2014, there was a 24-year-old woman who went in, got an MRI, and learned that she was missing her entire cerebellum. That's the lower back portion of your brain involved with physical movement and coordination. Now, she's got a completely normal life. She's a mom, she's graduated school, but she's always had jerky and clumsy movements. And now she has an explanation. Another example, in 2027, a 44-year-old man went in because he was having weakness in his leg, took an MRI, and they found that the spaces inside of his brain, which are always there for us, they're normal spaces, were so filled with fluid that it smushed his brain tissue out to the side, and he had a decrease of brain tissue by 50 to 75%. Now, this is the most drastic case we're going to see. And he was a normal functioning man. He was a parent. He worked as a civil servant. He did have an IQ of 75, which is lower than the average of 100, but it's not low enough to be considered mentally disabled. And so this is a perfectly normal person with 50 to 75% less brain than expected. Here's another example. Christina Sandhouse, when she was 7, she had a hemispherectomy. When we say ectomy, it means removing something, and hemisphere referring to one half of her brain. She had this hemispherectomy, and the reason that we do it mostly is because she had a seizure disorder. So we often do this in children. And so they took out the area of the brain that was causing trouble. And she is totally fine. She has some jerky movement. I have a video of her for you, so you can see Christina explain her experience in her own words. But she's a speech pathologist. She's totally working in a high-level job that requires a lot of cognitive ability, and she has half a brain. When we do these hemispherectomies, we've done follow-up studies. Here's another MRI where you can see half of the brain has been removed. And in the follow-up studies of 115 kids that had hemispherectomies, they found that 71% were seizure-free after, which is amazing because that's what they were trying to do, is get rid of the seizure disorders. 60% participated in mainstream schools with assistance. 70% had satisfactory speaking skills, and 83% could walk independently. So those are all really high numbers for removing half of your brain. Like, that's incredible. It turns out that we can compensate. So what used to be on the right side moves into the left and vice versa, which probably explains why Elise G was able to have such great cognitive language skills, even though she was missing her left temporal part of her brain, which is associated with speaking and language. I've got the study for you here, and I want you to pause the video and practice citing this in CSE format. The reason I'm asking you to do this is because your research project is coming up. It's due during week 9, and citations are a big part of your research project. CSE format is the Citizen for Science Educators format, and it's not citizen, it's criteria for science education format. But anyway, it's what biologists use, and it's what many scientists use. It's really similar to APA. So if you're comfortable with APA, it's pretty much the same thing. The format goes like this: author's last name, comma, first initial. You're going to do that for every author, and then you'll put a period. Then publication year, the year it was published, then a period. The title of the article, so long-term functional outcomes, period. Then your publisher information, the date you accessed it, and your DOI or URL. The DOI is the digital object identifier. This is pretty much a URL just for a research paper. I like using the DOI instead of a URL because it's often shorter. It will always take you directly to this paper. If you use a URL, you might use a URL from the online library repository. This is not the publisher, that's just the library that housed it. So make sure you feel comfortable with this. Do it right now, pause it. If you have any questions, email me, and then we'll move on. But please do practice your citations. If you want to change this format that I just told you into APA, the only difference would be that you'd put the year, publication year, into parentheses instead of a period. So it's name, date, title, publisher information, when you accessed it, and then how you found it, URL or DOI. Whether or not it's in parentheses or a period for the year, that's the difference between APA and CSE. So really similar formats. Now let's get into a nervous system. So we're going to go through how we evolved a brain, because the brain is like the highest organism level that we have for a nervous system. And we're going to start with cnidarians. Cnidarians have a decentralized nerve net, which means they have neurons throughout their body, but they're not organized and coordinating. Next, we're going to have neurons that are bundled together into fibers called nerves. So a nerve is a bunch of neurons together. The first example where we see that in the animal kingdom is echinodermata. That's going to be things like our sea star. Now, when we have nerves, we're going to take the axons and bundle them together, and then we're going to need to bundle all the cell bodies of a neuron together. When we bundle cell bodies together, we call it a ganglia. So here in planarians, our flatworms, we have central ganglia, which are the bodies of neurons all clustered together, and that can function as a brain, but it's not really yet a brain, so we call it a primitive brain. Now we still have nerves, which are the bundles of neuron fibers together. Next, we're going to have peripheral ganglia. That's going to be alongside with a central ganglia, and it's going to go down a ventral nerve cord. So a nerve, again, is a bunch of axons together. A ganglia is a bunch of cell bodies together. We see this in arthropods, like our wonderful bee here in the family Hymenoptera. Then we see complex brains, millions of neurons. We start to get some really interesting sensory organs associated with those brains. Bees have eyes too, but the protrusion of eyes right from a brain is pretty cool. We're still going to use nerves. We're still going to use ganglia. We just become more complex with more cells involved. That's what mollusks have, mollusks like this octopus. And then we have a full central and peripheral nervous system in vertebrates. The central nervous system is the brain and the spinal cord. Note that mollusks don't have spines, and so they do not have a spinal cord. And then the peripheral nervous system is everything outside the brain or the spinal cord. So all the nerves running down your extremities, down your cat's paws, and arms and legs, that is going to be your peripheral nervous system. We're going to have a functional brain, and we're also going to have ganglia alongside our central and peripheral nervous system, and nerve fibers, which are those axons and neurons. What is a neuron? Let's discuss. So here we have the overall cell body of the neuron. That's where we're going to find the nucleus. Inside here, if they showed it small enough, it would just be chock full of mitochondria. Mitochondria produce ATP, and your neurons need a lot of ATP, so they have a bunch of mitochondria in them. We're also going to have endoplasmic reticulum and vesicles and all sorts of goodies in here because this is a normal cell. Now, what's not normal about it is that there are protrusions from your cell to increase the surface area. Some of these protrusions are used to receive incoming information. We call those dendrites. Dendrite refers to the branching pattern. Then we're going to have a long branch coming off one side of your neuron. This is called the axon. And at the end of your axon, you're going to have more branches, but now we call these synaptic buds or synaptic terminals. And then a synapse is where this neuron connects to the next neuron. So these are the dendrites of a different neuron. We have motor neurons, which help us move our muscles. We have sensory neurons, which take in information from the outside world. That's things like touch and pressure and pain, but also temperature and sunlight and smells and tastes. All that is sensory information. And then we have neurons that connect the two. This is information in, this is information out. This is your connector, your interneuron. In this picture here, we have a couple things that we didn't talk about yet, and that is these weird blue structures. These are different types of cells. They're called glial cells. I have that coming up, so don't feel like you have to write it down right now. Their names are oligodendrocytes. Such a great word. What these glial cells are doing for this neuron is creating an insulation bundle around the axon. This is called myelin. Myelin is going to help your axon move the information from the cell body to the receiving cell quicker. So myelin is like insulation around a wire. It just keeps the electrical impulse inside. And we know that our ultimate goal with a neuron is to send electrical impulses from one cell to another. We also like to classify neuron based on their dendrite location. So if the dendrites are over on one side only, we call it unipolar. If it's on both sides, it's bipolar, bi for two. If it's on all one half of the body, and then we have that long axon like we draw usually, this is our stereotypical neuron. It's multipolar because now we have multiple sets of dendrites. And then if we have what looks like a bipolar neuron, but the cell body is pushed off to the side, it's called pseudo-unipolar because there's one branch off the cell body, just like unipolar, but it has two further branches for the dendrites. So it's not bipolar, so it's false unipolar. That's what pseudo means, is false. We also describe neurons based on their overall shape. This one kind of looked like a pyramid to the first person that described it. So it's pyramidal cells. We find these a lot in the cerebral cortex, which is the main part of our brain that we use for thinking and rationalizing and feeling. We also have Purkinje cells. Those are in the cerebellum, back in the area that we use for coordinated movement. I love Purkinje cells because they're so gorgeous and they're so highly branched. A lot of interconnections between your neurons there. And then we have a special type of neuron associated with our olfaction, which is our sense of smell. Each one of these neurons here that's shown in this epithelial layer, this is simple columnar epithelium. The neurons poke through and they have their receiving end out in the mucus of your olfactory organ. This is where they will bind to the sense that you're smelling. And then they will be activated by that smell particle binding to them. So it's like a physical binding. And then they send that information up right into your olfactory bulbs, which are extensions from your brain. So this is a very short, direct connection. And that's why odor is so strongly emotional for a lot of us. You can smell something and immediately be transported back into a feeling or a memory. Like anytime I smell pumpkin pie, I think about my mom. She loves pumpkin pie and I love pumpkin pie. So smelling pumpkin pie right into the part of my brain that makes me think about my mom. How do neurons work? Their whole goal is to send electrical signals. So they're going to have one part that's information in, process it, and then information out. So here is my neuron. Up at the dendrites, this is information in. I'm going to get this information from something like a odor particle or a temperature receptor or a pressure receptor, or I'm going to be hearing something, whatever it is, I'm taking information in. That information goes in the dendrites, through the dendrites, into the cell body. Look, we can see all the goodies in there now. So those are the mitochondria and the endoplasmic reticulum. Here's your nucleus storing the DNA. And in the cell body, that information is going to be processed. And when it's processing it, it's really asking, is this important enough to send on? And if it is, then it will send it on. If not, the signal will kind of just die out. So if it's important enough to send on, it'll go through this tapered region of the neuron called the axon hillock. And then it will go through the long tail called the axon. This is a single direction. It cannot go back up the axon. We'll talk about why in a second. Once it starts to send it through the axon hillock, it's like a yes or a no signal. So it's been switched. It's going to go. The signal will now run down the axon, and it will come to the body or the dendrites of the next cell. And it will share that information with this cell. And now this cell gets to do the same process. It received the information, it's going to process it, send it on, yes or no. If yes, it sends it on its axon down to the next cell. So some things to note. This is the presynaptic cell, and this is the postsynaptic cell. What is a synapse? A synapse is any location where two neurons are talking to each other. So this is before this cell, so it's pre-synapse, and this is post-synapse. Since it's a singular direction, you don't go backwards up the axon. It always has an orientation of pre-synapse post. We also are showing myelination on this axon. That myelin works as an insulator, so it helps the information go down that axon more quickly. I mentioned that it's a one-way process. Let's discuss why. So here I am. This is my neuron. These are my dendrites. This is my cell body. Soma means body. Remember, somatic. Then we have our axon and our axon terminals, which will be the synapse to the next cell. When this neuron is sending an electrical impulse, it takes the charge inside the cell and it briefly flips it. So the resting charge of your neuron is negative. There are a bunch of proteins in there. They're negatively charged. When it is told that it's going to send a signal, it gets a chunk of positive charge moving down the axon. So here we could say that this is depolarized because we've changed the charge. This positive piece of charge is going to start up at the axon hillock when it decides to send the signal. This pulse of positive charge moves down the axon. So see, the green is moving down the axon. And after it moves through, it takes a minute for your neuron to be able to repolarize. Polar means that the charges are separated. So positive and negative are separate. Just like on our Earth, we've got a magnetic north and a magnetic south. Those are our poles. Same inside of a cell. Inside the cell is negative, outside is positive. That's polar. So we are reestablishing our negative charge inside our neuron, and it takes a minute to do this. So you can't go backwards. It's not ready yet. You couldn't make this positive going in the other direction. Because of that, the positive charge, that little pulse of positive, moves forward because it can't go back. It can't do it again. And so it just creeps really, really fast. It's not a creep. It shoots really, really fast down the axon toward the postsynaptic cell. And as it repolarizes, it eventually returns to resting and it can send another message. So it doesn't permanently alter the charge. It's just a temporary flux, and that positive flux moves down the axon to the synapses. How does this work? It's all about membrane potential. So membrane potential is the difference between the inside and the outside of the cell. The membrane is like the gatekeeper, and it's keeping negative in, positive out, until it's ready to send a signal. We've actually measured this. We've taken a little voltage meter, and we've stuck one electrode into the extracellular fluid, and then we stuck the other electrode into the cytoplasm, and we found that the difference is about negative 70 millivolts. So in negative 70 millivolts, your cell is just resting. Your axon is not doing anything. Inside of your neuron, you're going to have a lot of potassium. It's got this positive charge. And outside your neuron, you're going to have a lot of sodium. It's got a positive charge. What we're not showing is all of the proteins inside your axon that gives it a net negative charge, negative 70 millivolts net. So we're showing you ions, but overall, the resting state of a neuron is more negative inside. We have two players in this. First, let's orient ourselves. Here is my plasma membrane. So it's the phospholipid bilayer. Here are the phospholipid heads and tails. I have proteins embedded in my plasma membrane. One of them is a potassium channel. It's closed right now, but when it opens, it's going to let potassium out. One of them is a sodium channel. It's closed right now, but when it opens, it's going to let sodium in. And then we're going to have a sodium potassium pump. This sodium potassium pump is going to revert everything that we've done. So this sodium potassium pump is going to be important to putting it back. Note that the sodium potassium pump uses ATP. So this is costly. Every time you use a neuron, you have to pay to put it back into the resting state. And that's why your nervous system can use up to a third of your calories per day. Like, it's costly to use this big, beautiful brain that you have. So let's do it. Right now we're resting. Nothing is happening. Your dendrites are getting a message. They've decided that they need to send this message on. They're now going to create a temporary charge that's positive, and they're going to throw that down the axon to the next cell. That's how they send on their signal. We call that propagating a signal. This temporary flux of positive charge moving down the axon is called an action potential. So we are creating an action potential. How do we create it? We're going to open that sodium channel. So now the sodium channel is open. All of this positive charge is going to rush in, like you've opened a can and you're pouring positive into your cell. And that positive is going to rush into your axon, and it's going to change your charge from -70 to briefly about, almost 40 positive. So we've gone from negative inside to now positive inside. That's our electrical change. Your cell notes that it's letting the sodium. So now it's going to snap that sodium closed, and it freaks out. It's like, oh my gosh, I'm much too positive. I need to get out some of this positive charge. It would make sense to just push out all that sodium that you let in, but your cells don't do that. Instead, what they do is they take their potassium that was already in there and they start throwing the potassium out. So your potassium channel opens, you throw the positive charged potassium out, and that allows your cell to drop back to resting almost. You kind of go too far. We call this the overshoot. So you become too negative. And now you're going to use your sodium potassium pump to put everything back. We can graph this. Here I have membrane potential. That's a measure of the positive and negative on the inside and outside of my cell. I have time in millisecond. 1 millisecond is 1,000th of a second. Here I have -70 millivolts. That's the resting potential. It's about -70 millivolts inside of a cell when it's not being used. I say cell, but I mean a neuron, inside of a neuron. Now your cell, your neuron, is going to decide to propagate an action potential. It's going to open those sodium channels. That sodium is going to rush into your cell. It's going to create a positive charge. So we're getting up almost to +40. So we're increasing the charge. Because we're increasing the charge, we are ruining the separation of positive and negative. So we're depolarizing it. Then your sodium channel is going to snap closed. Your potassium channel is going to open, and you're going to push potassium out. Potassium has a positive charge. So as you remove the positive charge, you become more negative. This is repolarizing. We're separating the charges again. You're going to push out too much potassium. That's the undershoot, and we always do this. And after your undershoot, you're going to use your sodium potassium pump to return to resting potential. Let's look at this another way. Here I have that graph we just described. Resting state at -70, depolarizing to +40, repolarizing, but you go too far, you're undershoot, and then back to resting state. We can organize each of these with something that's happening. So here in my resting state, I have my membrane, that's the yellow, so this is my plasma membrane. Outside of my cell is more positive, inside my cell is more negative, so I'm polarized. The first thing I'm going to do is open the sodium channel. When I open my sodium channel, positive is going to rush in and I've depolarized. Now the outside of the cell is more negative and the inside is more positive. We've flipped the charge. So we're taking a little packet of positive and we're pushing it down the axon. We'll snap close that sodium channel, and then we'll remove the positive potassium from our neuron. So we push out this one. That's going to repolarize us. We're going to turn back so that the outside of the cell is positive and the inside is negative. We tend to go a little too far. We shoot out too much potassium. We'll close this gate and we'll use that sodium potassium pump to reestablish the ions into where they ought to be. All right, now let's see what happens after that signal is propagated down the axon. And we come over here to the synaptic terminals and synaptic buds. They're called buds because they're often a little bit swollen. So here, this is the axon end. When the presynaptic cell wants to talk to the postsynaptic cell, they don't actually touch. There is a gap between them. This is called the synaptic cleft. And in this synaptic cleft, they're going to communicate with chemicals called neurotransmitters. So the action potential coming down this axon from this cell to the next cell is going to trigger the release of these neurotransmitters using vesicles and exocytosis. Those little chemicals released into this synaptic cleft, the space, they will be received by the postsynaptic cell only if it has receptors to it. If it doesn't have receptors, then it's just falling on deaf ears. It doesn't get heard. But if it does have receptors, that can trigger a depolarization event in the membrane of the postsynaptic cell. So without physically touching, these two cells can communicate. Here's another visual, some more vocab. The action potential reaches the presynaptic terminal, so it comes all the way down the axon into this synaptic terminal or synaptic bud, this swollen part of the axon at the end of it. As it gets all the way through here, the depolarization of this terminal membrane allows calcium to flow into the synaptic terminal. And that will create little vesicles full of neurotransmitter. These vesicles will fuse with the plasma membrane. They will exocytosis, and they will eject their neurotransmitter. Here's these little neurotransmitters in the space between the two cells called the synaptic cleft. This cell here is the dendrite or the cell body of the postsynaptic cell, and it's going to have receptors. If those receptors bind to the neurotransmitter, it could create a depolarization in this cell. So the message from this cell gets received chemically into the next cell, and that allows an action potential to form, and then you can propagate the signal down the next axon. So these two cells communicate. We have two systems in vertebrates, the central nervous system and the peripheral nervous system. The central nervous system is going to be your brain and your spinal cord. I've got two diagrams to explain this. We'll look at the left one first. Your brain and your spinal cord are going to be found in a protective layer of bone, either your cranium or your vertebral column. This is why it's in vertebrates. And then all of the nerves and ganglia that are found outside of those two regions, those are called the peripheral nervous system. Your peripheral nervous system is divided into two parts. One is somatic. Somatic is going to have sensory input, so temperature, pressure, pain, sound, smell, taste, and it's going to have motor output. So movement, squeezing of your organs, remember smooth muscles, unconscious, elevating and decelerating your heart rate, all that kind of stuff is somatic. Now your autonomic nervous system, this is subconscious. This is something that is more important to keeping you alive long-term, and it's divided into two parts, sympathetic and parasympathetic. Your sympathetic nervous system is the fight or flight or freeze response. So that's going to be response to stressful events. And this is stressful events like you're almost in danger. Now, your other side, your parasympathetic nervous system, We call this the rest and digest. This is when you're calm, when you're safe, when you're going to feel sleepy. That's parasympathetic. Two halves of our unconscious nervous system. Here's another diagram. Our central nervous system, brain and spinal cord, and our peripheral nervous system. Those are the two locations of where we keep our neurons. Our two locations communicate, so I like that they show these arrows going back and forth. The peripheral nervous system is where we take in information, bring it to the brain for processing, and then your brain will respond back into the peripheral nervous system. So that little exchange is a nice way to visualize that these are integrated. It's not one or the other, it's always both because we're going to use both constantly. We send information into our peripheral nervous system from the sensory division. We respond with the motor division. And we also have that autonomic nervous system or the somatic nervous system. The somatic nervous system is going to be sensory and respond to muscles. This is voluntary. Our autonomic nervous system, this is not voluntary. This is all subconscious. I think automatic, autonomic, those things go together in my head. The sympathetic division prepares the body for emergencies, and the parasympathetic controls you when you're not having an emergency. Glial cells are incredible. Glial cells are all the cells in your nervous system, so in the CNS and the PNS, that are going to support, protect, and take care of your neurons. Glial cells outnumber your neurons 10 to 1. So you have 10 times more glial cells than you have neurons. And they have a variety of function. We have a few more in the central nervous system. So in the central nervous system, we have oligodendrocytes, which, what a fun word. Oligodendrocytes. The word cyte means cell. These are the ones that form myelin around the axon. So here are your oligodendrocytes. They're connected. This is their cell body. And these are protrusions of their cell that wrap around that axon, allowing like a lipid barrier that makes your axon move more quickly. And by move more quickly, I mean the action potential can move more quickly through your axon. Then we have astrocytes. Astro means star, so someone thought these looked like little stars. These are going to provide nutrients and structural support for your neurons. They are going to talk to ependymal cells. Ependymal cells produce cerebral spinal fluid that fills all of the spaces inside and around your brain. They're good for cushioning, but they're also really important because they allow nutrient exchange and removal of waste. So cerebral spinal fluid, CSF, is very important for keeping your system healthy. And then we have microglial cells. They're smaller, hence the micro. These are going to be looking for pathogens and dead cells, and they will remove them. In the peripheral nervous system, we're going to have two types of glial cells. We're going to have Schwann cells, which create myelin sheaths. Now, the first time I heard about Schwann cells, they were described as wrapping around the axons, jelly roll style. So I love that visual. So they're just rolled around like how you would burrito a baby, just roll that baby up in a blanket. That's how the Schwann cells support the axon. And they are lipid-based, so they create that insulating layer to allow the action potential to move through your axon more quickly. And then we have satellite cells. Satellite cells are going to provide nutrients, they're going to remove waste, and they're going to create structural support. So that's all these little red cells hanging out on the cell body of this pseudounipolar neuron. Check out that human brain. The human brain has about 86 billion neurons connected with 100 trillion synapses. And those numbers are so big. Like, those are kind of wild. It's one of the most complex objects in the known universe. We still don't really understand it very well. It is highly vascular. So on here, you can see all the brown. That's actually blood that's coagulated into capillaries. but lots of blood is supplied to the brain because we need lots of oxygen and we need lots of nutrients. We need lots of ATP. A fresh brain is going to be very pink, very red because of that blood, and it's going to have the texture of flan. So it's really soft. It's often described as jello, but it's a little bit less jiggly. So it's just easy to damage, easy to bruise. Luckily, we can protect our brain, and we protect it with 3 layers of meninges. So meninges are going to be fibrous layers around the brain. The first one that's right on top of the brain, so it's adhered to the surface of the brain, it's called the pia mater. Mater means mother. So we have pia mater, which means gentle mother. So the gentle mother is right on top of your brain, protecting it. This is important for nutrient exchange and waste removal. Then we're going to have a layer called the arachnoid mater. Here's a little protrusion of it. The arachnoid mater is going to be kind of cobwebby, and so it's the spider mother. That's what its name means, arachnoid spider. And this is something we can see during brain dissections. It looks wispy and cobwebby. And then we have a nice thick layer. That's the dura mater. Dura means tough. So this is the tough mother. And it is dense, like it is like a plastic bag surrounding your brain. We can see it in some videos later, but it's going to hold your brain in place by going down in between the 2 hemispheres and then down between your cerebellum and your cerebrum. So like a little tint around that portion of your brain. And it's going to hold everything in position so that when I whip my head around, my brain is not swirling in the fluid that is in there. We define our brain regions based on the bones that is closest to them. So the frontal lobe has the frontal bone, the temporal lobe has the temporal bone, parietal lobe has the parietal, occipital has the occipital. But each of these pieces of the brain has different functions. So we divide into right and left, and we also divide our biggest part, our cerebrum, from our cerebellum. Here we have another visual of the areas of the brain. Our frontal lobe has a couple of cortexes. Cortex means region. This area here in the prefrontal cortex is Brocaw's area. That's the left temporal. That's associated with speech. Then we also have motor areas. So our frontal part of our brain is associated with motor movements. This is the primary motor cortex. We think this is the region of the brain that does the most motor movements. Abutted next to it is the sensory cortex, primary sensory cortex. So that's going to take information from our senses. Every time we see a ridge, it's called a gyri. Gyrus is singular. And the grooves in between them, those are the sulci. Singular would be sulcus. Here we have Wernicke's area. That's going to be involved in language comprehension. And this large region right here is the general interpretive area that allows us to take all of the sounds and sights and things we want to say and interpret them together. In the back of your occipital lobe, you're going to have your primary visual cortex. And then your cerebellum, which is not being highlighted here, but that's a lot of motor cortex. Now we have cut the brain into a frontal section. So we've removed the front and we're looking in on it. In our brain, we have these spaces. Those are going to be filled with CSF, cerebral spinal fluid. In our cerebellum, in our cerebrum, the gray matter is going to be on the outside and the white matter is going to be on the inside. The gray matter is made of a lot of cell bodies and dendrites. The white matter is white because it's lipidy, and it's full of lipids because that's where myelin is. So these are going to be full of axons. Myelin sheaths are wrapped around axons. Here we have our brainstem. This is part of our cerebellum. You can see the protrusions, the gyri really well. And then in between them, these deep grooves, those are your sulci. Here is our division between our right and our left hemisphere. So that's our hemispherical fissure. And then we have fibers connecting the right and left half. That's called the corpus callosum. So this is fibers connecting the right and the left half of the side. And here we have a string of axons connecting the top and the bottom of our brain, referred to as the cerebral cortex. This is our ventricles. Our ventricles are the empty spaces in our brain. And these empty spaces are filled with cerebral spinal fluid. I love this visual of it moving because you can see the overall structure of them. We need cerebral spinal fluid on the inside of our brain because that's how we're getting oxygen to the tissues. That's how we're removing waste. That's how we're getting nutrients. So it's like a constant bath, refreshing the waters. So we're going to take in all the good stuff and remove all the bad stuff. These ventricles can be too full or not full enough, as we saw in the beginning of the lecture. The ventricles are described as lateral ventricles. We have two lateral ventricles. They're the ones that go out to the sides. Then we have a third ventricle, which is where they connect. And then a tube that drains down, that's called an aqueduct. So we have the central aqueduct, another space, the fourth ventricle, and then this one, the central canal, that's going to go all the way through your spinal cord. There's a tiny little hole that we're going to talk about later, but that allows us to keep our cerebral spinal fluid moving through these tissues of our brain. All right, now let's discuss the limbic system. The limbic system is not one thing. It's a whole bunch of regions of the brain together, and they're not really distinct to some of them. Some of them are, you can like point to it, but others are kind of like a general area. The limbic system is involved in motivation, emotion, learning, memory. You use your limbic system in this class all the time. And it's composed of the thalamus, the hypothalamus, so that's these two regions right here. You're also going to have your hippocampus, that's this kind of purpley region, and your cingulate gyrus. Hard for me to say, go ahead and say it to yourself. Latin is fun. And then you're also going to have your hippocampus. So that's often referred to as your amygdala body. Right there, you have all your fun things together. Our diencephalon is the structure of our brain that's right in the center. It's surrounded by those ventricles, and it's composed of the thalamus and the hypothalamus. Your hypothalamus and your thalamus are below this purple region, which is your connectors between the two sides of the brain that we discussed. And then you're going to have your brain stem. Your brain stem is the midbrain, the pons, and the medulla oblongata together. So it's this portion of your brain that's going to connect your cerebellum and your cerebrum to your spinal cord. Now, your brain stem is really important for keeping up your heartbeat, your breathing rate, a lot of your vital organ systems. So that's a large part of your brain keeping you alive. It's sometimes referred to as your lizard brain because it's pretty much the same in you and a lizard in its functions. Now we're looking at the spinal cord cut into a section. So this would be like if we cut right here and looked down into your spinal cord. Note we have a reversal of the gray matter and the white matter. So now all of the axons with myelin are on the outside, and the cell bodies that do not have axons, the gray matter is on the inside. So we have a reversal. This structure is often described as the butterfly of the spinal cord, or a letter H, but I like the butterfly a little bit better. And there would be a hole right in the center that would be connected to your ventricles that would allow CSF to run through your spinal cord. Your spinal cord is going to take all the information in from your body, send it to your brain, allow you to process it, and then whatever your brain decides, that information out will go through your spinal cord as well. Your spinal cord controls your motor reflexes. So if you touch a hot pan and you pull back your hand, that's actually done before it gets to your brain. It's decided by your spinal cord because that's maintaining homeostasis. If there's pain involved, your body's going to want to respond quickly because that means that there's something bad. We call that a reflex arc. So here I have a needle or a pin poking through skin. So this would be a pain response. Imagine I just poked with a needle. In our skin, we have sensory neurons. Those sensory neurons will receive that pain response. It'll cause a depolarization event. That depolarization event will run down the axon of this neuron through a nerve. Here's a bunch of cell bodies in my nerve called ganglia, but through this nerve in the peripheral nervous system to the spinal cord. So now we're in the central nervous system. We'll have an interneuron that's going to say, hey, you were just stabbed by something, you should move away from that pain. And that interneuron will send the signal to move your hand away from the pain through another neuron that will go through this peripheral nervous system nerve, through the axon to the muscle, and will make you respond by removing your hand from the pain. So we call this a reflex arc. We have information in, interneuron, response out. This is done fully in your spinal cord. Your spinal cord doesn't need to consult your brain. If it's something that your brain needs to know, the information will be sent up. That's like the big boss. If you can fix it yourself, you don't alert your boss. But if you can't fix it yourself, then you tell the boss. Our peripheral nervous system, we said, was divided into the autonomic nervous system and the somatic nervous system. The autonomic nervous system is going to provide body controls without your own input. So this is unconscious. And your somatic nervous system is going to be taking information from your skin and your muscles, sending it to your CNS, and getting some kind of response to it. This you often are aware of. So you're aware when you're uncomfortable and you need to move, or when you're a little cold or a little hungry. Those things you're conscious of. There we are. Your autonomic nervous system has pretty much opposite effects. So we said that the sympathetic nervous system is stress. This is, I'm scared, I need to run, I need to fight, I need to freeze. The example that I like to think of is like when you almost get into a car accident and you feel that like rush of adrenaline and fear, that's your sympathetic nervous system getting activated. Or if you've ever leaned back in a chair and it didn't stop and you fell over, or you're going down steps and there's one more step than you think and you have that moment of, oh gosh, I'm falling. All of that is sympathetic nervous system, but this is also like rage. So in your sympathetic nervous system, and I remember sympathetic because when scary things happen, we feel bad. So we feel sympathy for the sympathetic nervous system. You're going to divert your energy in your body to keeping you alive. So if I'm in a dangerous situation, I'm going to want to dilate my pupils to let more light in. I'm going to inhibit saliva because I don't need to eat right now. I just need to stay alive. Going to increase my heart rate, get more blood to all my muscles because I'm going to need to like run. I'm going to dilate my bronchi so that I can have more oxygen flowing into my body so I can run or fight. I'm going to inhibit digestion, but I am going to break down glycogen so that I pump more sugar into my bloodstream so I can use it. I'm going to secrete my stress hormones, adrenaline and noradrenaline. So those are the ones that make you feel kind of scared, feel anxious. I'm going to inhibit my bladder. So it's not important to urinate right now. I'm going to function on just staying alive. The opposite is when I'm not feeling stress. So if this is fight or flight, parasympathetic is rest and digest. So this is when I'm at home at night with my cats and my book and a snack. That's my parasympathetic system. It's not important that I let in a bunch of light. So I'm going to constrict my pupils. I'm going to salivate. I'm going to get hungry. I'm going to want a little snack. My heart rate's not going to be racing. I'm not going to be breathing heavily. I am going to be interested in digesting. Digesting takes a lot of energy, but I don't need that energy to stay alive and like run and fight. So I'm going to stimulate digestion, stimulate bile secretion, and allow my bladder to become aware when it's full. All right. That was our nervous system and our brain. I hope it was interesting. Email me anytime with questions, and I hope you have a great day. Bye.